I want you to do something for me right now. Just take a deep breath,
Yeah, go ahead.
fill your lungs. Now let it out. That oxygen you just breathed in, it is the absolute non-negotiable fuel keeping you alive right this very second.
Right.
But here is the crazy part. That exact same oxygen is simultaneously rusting your cells from the inside out.
It really is. It's a biological double-edged sword.
Exactly. And it is at the very core of why we age. Welcome to today's deep dive. We are thrilled to have you with us as we explore the invisible, lifelong war your cells are fighting against oxidative stress.
And you know, science is finally uncovering ways to actually turn the tide in that war. Today, our mission is to examine how researchers are literally hijacking the dormant secrets of embryonic stem cells to fight oxidative damage.
Yeah.
And incredibly, reverse the aging process itself in living tissues.
We have a really fascinating stack of sources to unpack for you today. We're looking at a foundational 2003 paper from Nature Cell Biology that solved a massive, decades-old mystery.
Right, about why mouse cells age so differently than human cells.
Yep. Then we are jumping into a huge breakthrough from September 2025 on using partial cellular reprogramming to restore vision and actually reverse aging in the eye.
And finally, we will touch on a brief 2026 update showing how these exact same factors are being used to heal joint injuries.
It's going to be a lot of fun.
We will be bridging the gap between the microscopic details in these papers and the broader implications for human longevity. The goal is to show you exactly how these cellular mechanics translate to the bigger picture of how we age.
And how we might stay healthier longer. Okay, let's unpack this 2003 Nature Cell Biology paper because it starts with a genuine biological puzzle.
It really does.
There is this concept called replicative senescence, which is basically a cell's forced retirement plan.
Right, because cells have a strict built-in limit on how many times they can divide before they simply stop functioning and senesce.
And in human cells, we track that countdown using telomeres.
Which are the protective caps at the ends of our DNA strands.
You can think of telomeres like those little plastic tips at the ends of your shoelaces. Every time a human cell divides, those plastic caps get a little bit shorter.
Right.
And when they wear down completely, the shoelace frays, the cell stops dividing and it retires.
But here was the mystery. Mice actually have incredibly long, highly stable telomeres.
Right.
So by all biological logic, their cells should keep dividing in a lab dish much, much longer than ours.
But they don't.
No, they don't. When scientists put mouse embryonic fibroblasts,
Or MEFs.
yeah, MEFs,
Yep.
when they put them into a standard petri dish, they stopped dividing and senesced way faster than human cells.
Which makes no sense. The answer to that puzzle came down to a fundamental oversight in how we culture cells in the laboratory.
Exactly. The standard condition for growing cells in a laboratory incubator uses 20% oxygen.
Which is just the atmospheric level of the air you and I are breathing right now.
But inside a living mammal's body, tucked away deep in our tissues and organs, the physiological oxygen level is actually only about 3%.
3%. That is a huge difference.
Massive. So the researchers took these mouse cells and cultured them in 3% oxygen, matching their natural physiological state.
And the results were a massive aha moment. Everything changed. The cells grew faster, they reached much higher densities, and the real shocker,
They were senesced.
they didn't hit that forced retirement wall at all. They just kept growing continually for over 60 population doublings.
The researchers realized the mouse cells in those standard incubators weren't dying of old age in the petri dish. They were dying of severe oxidative stress.
It was essentially a fatal case of culture shock.
Right. And to actually quantify this shock, the scientists used something called an FPG Comet assay.
Which is a technique that visually measures oxidative damage to DNA.
Yes. And the results were stark.
Mm.
In 20% oxygen, the mouse cells accumulated 3 to 4 times more oxidative DNA damage than they did in the 3% oxygen environment.
But the comparison between mice and humans is where it gets really wild. The mouse cells sitting in 20% oxygen have 3 times more DNA damage than human cells sitting in the exact same 20% oxygen.
If we connect this to the bigger picture, this tells us something profound about human biology.
Oh, absolutely.
Human cells possess a vastly superior, deeply ingrained ability to prevent or repair oxidative DNA damage compared to mice.
We are biologically built to handle atmospheric oxygen in a way they simply are not.
Exactly. This fundamental biological difference likely explains a lot of the major variations in cancer incidences and the overall rate of aging between our two species.
And just to prove that DNA damage was the true culprit, the researchers also tested cells from special mice genetically deficient in DNA repair.
Specifically mice missing a protein called Q80.
Right, and those cells senesced rapidly, even in the safe 3% oxygen environment. So it really all comes down to the DNA damage caused by oxidative stress.
The massive takeaway here isn't just about mice, though. It's the realization that for decades, our standard laboratory environments have literally been causing premature aging in our test subjects.
The air in the room was completely skewing our understanding of cellular aging.
Yes.
Knowing that atmospheric oxygen is essentially a slow poison for cells in a petri dish, it makes total sense that the parts of our living bodies exposed to the most oxygen and light take the hardest hit.
Which brings us to the eye.
Exactly. That brings us to our 2025 source regarding a massive breakthrough in the human eye.
There is a layer in the eye called the retinal pigment epithelium.
Or RPE.
Because of the constant bombardment of light and high oxygen demand, the RPE takes an absolute beating.
And this constant stress leads to age-related macular degeneration, or AMD. If you know anyone who struggles to read a menu at a restaurant because the center of their vision is blurry, you are seeing the real-world impact of the RPE failing.
The RPE acts as the daily maintenance crew for your photoreceptors. Over time, that relentless oxidative load causes the RPE to accumulate damage and eventually break down.
So what can we do about it?
Well, the 2025 paper explores a highly advanced intervention, partial epigenetic reprogramming using the Yamanaka factors.
Right, these are specific genes, OCT4, SOX2, and KLF4, often abbreviated as OSK.
In the biology world, they are famous for being able to turn a mature adult cell all the way back into a blank slate embryonic stem cell.
But the goal in this research isn't to turn the eye cells completely back into stem cells, right?
No, it's partial reprogramming. They just want to turn back the clock a bit and rejuvenate them.
To do this, the researchers used a harmless virus, specifically AAV2, essentially as a microscopic delivery truck, to drop off the OSK genes directly into the subretinal space of aged mice.
We are talking about 15-month-old mice.
Which is senior citizenship in mouse years.
Right. They measured visual acuity using an optomotor response assay. Within just 4 weeks, the older mice's vision improved significantly.
And by 8 weeks, their visual acuity was completely comparable to 3-month-old young mice.
It's incredible. When the OSK therapy enters the RPE, it acts like a cellular renovation crew. The researchers observed that OSK actually restored youthful melanin granules,
which are critical for absorbing stray light and protecting the eye against further oxidative damage.
Exactly. It also repaired mitochondrial cristae. Those are the energy-producing folds inside the mitochondria. And it significantly reduced abnormal lipid deposits.
And it even worked on a genetic model of AMD. They tested it on mice carrying a human risk gene for AMD, the CFHHH mice.
And they put them on a high-fat, high-cholesterol diet to really stress their eyes out to the maximum.
The OSK therapy still worked. It significantly reduced abnormal cell multinucleation, which is a major glowing red flag of cellular stress in the eye.
But there's a catch.
There's always a catch.
While the OSK treatment rejuvenated the RPE and restored vision beautifully in the short term, prolonged expression of these genes beyond 8 weeks caused detectable toxicity to the photoreceptors. Furthermore, when they analyzed the cells, they found that OSK paradoxically increased the transcriptomic age of the cells in some specific ways.
Okay, let's break down what transcriptomic age means for a second. Even though the cells looked younger structurally, their transcriptomic age, meaning the overall genetic instruction manual they were actively reading from, was actually showing signs of accelerated aging.
Right, it was activating pro-aging interferon and mTOR signaling pathways.
Because the Yamanaka factors are blunt instruments.
Very blunt. They change the expression of thousands of genes all at once. You can't just leave that stem cell factory turned on forever.
Right. If you leave the renovation crew in your house for too long without supervision, they don't just fix the kitchen, they start knocking down load-bearing walls and ripping up the plumbing.
Great analogy. The researchers realized they needed to fire the general contractors and just hire the specific plumber doing the actual work.
They needed a safer, more precise tool to get the benefits of OSK without the systemic toxicity.
So the researchers set out to find the specific downstream effector, the exact molecular tool OSK was using to fight the oxidative stress in the RPE.
And what they found completely overturned existing assumptions.
It did. Previously, science thought OSK did its healing work through a process called DNA methylation, which requires an enzyme called TET2.
That is the established mechanism for how OSK heals other parts of the eye, like retinal ganglion cells.
Right.
Here's where it gets really interesting. In the RPE, the researchers discovered a completely non-canonical pathway. The protection OSK provides to the RPE is entirely TET2 independent.
And it is highly dynamic. Remarkably, it leaves absolutely no permanent epigenetic memory.
Meaning the moment you turn the OSK genes off, the oxidative protection vanishes instantly.
Exactly. This transient nature suggested OSK was activating a very specific temporary defense mechanism. To isolate it, they ran a massive transcriptomic screen,
They looked at massive data sets across human cells, mouse cells, and fibroblasts.
trying to find which specific genes OSK was consistently turning on across all of these different types to fight oxidative stress.
And out of all that overwhelming data, one single gene emerged as the undeniable hero.
GSTA4.
GSTA4 is a detoxifying enzyme. Its primary job in the cell is to clear out a very specific, highly toxic molecule called 4-HNE.
Yes.
You can think of 4-HNE as a toxic byproduct of lipid peroxidation. It is the cellular equivalent of fats going rancid or spoiling inside the eye over time.
As the RPE ages and takes damage from light and oxygen, this rancid 4-HAE accumulates progressively. It is deeply toxic to mitochondria and tears apart cellular structures.
Think of 4-HNE like a toxic cellular exhaust that builds up in the engine of your eye over a lifetime of use.
And GSTA4 is the biological catalytic converter that clears it out.
So the researchers wanted to know if this catalytic converter was powerful enough to run the show on its own.
Right, so they overexpressed just GSTA4, completely leaving the blunt instrument OSK Yamanaka factors out of the equation.
And it was fully sufficient to completely protect mitochondrial function from severe oxidative toxins, like sodium iodate, in a petri dish.
But they didn't stop at the petri dish.
No, they didn't. When they used that AAV delivery truck to deliver just GSTA4 to living, aged mice, it actively cleared out that toxic 4-HNE exhaust.
It restored their vision.
And when they analyzed the RPE cells afterward, they found that GSTA4 reduced the transcriptomic age of those old cells by 8.5 months.
In mouse terms, 8.5 months is cutting their biological age entirely in half.
Wow.
And it achieved this staggering rejuvenation safely, completely bypassing the load-bearing wall toxicity issues associated with leaving the OSK factors turned on.
So if GSTA4 is this incredible age-reversing catalytic converter for our cells, why does our body ever turn it down in the first place?
That's the million-dollar question.
Why isn't this biological fountain of youth just left in the on position all the time as we age?
What's fascinating here is that the answer lies at the very beginning of life, during early embryogenesis. When an embryo is first developing, it is essentially a ball of highly active, rapidly dividing stem cells. During this early phase, both the OSK genes and the GSTA4 enzyme are firing on all cylinders.
Throwing up a massive shield to protect those precious early cells from any oxidative damage?
Exactly. However, there is a crucial developmental milestone called mesendoderm differentiation.
That is basically the moment when a blank slate stem cell has to pick its career path. It has to decide if it is going to grow up to be a muscle cell, a bone cell, or an organ.
And to trigger that differentiation, the developing embryo actually needs a sudden surge of reactive oxygen species, or ROS.
So it needs the stress.
Yes. It requires a targeted burst of oxidative stress to act as a chemical signal telling the cells it is time to change and specialize.
So if GSTA4 stayed turned on, its powerful antioxidant shield would neutralize that vital developmental signal, and the embryo wouldn't develop properly.
Exactly. So the body sharply shuts GSTA4 down to allow growth to happen.
This brings up a concept in evolutionary biology called antagonistic pleiotropy. It is a fancy term, but it basically means that a genetic trait that is highly beneficial when you are young can become actively detrimental to you when you get old.
Evolution really only cares about getting you to reproductive age.
Right. Turning off the GSTA4 antioxidant shield is totally necessary for you to develop as an embryo. But decades later, because that shield was turned down and left off, that toxic rancid fat exhaust is allowed to accumulate,
leading to things like macular degeneration and aging.
The OSK therapy, and now the much more precise GSTA4 therapy, is simply reaching back into our cellular code and reactivating a dormant embryonic shield.
And this pathway isn't just an eye-specific quirk. The 2026 source fragment we have notes that researchers are now locally delivering these exact same OSK factors to mitigate osteoarthritis and cartilage fibrosis in joints.
That is huge.
It demonstrates that this oxidative resilience pathway has massive whole-body potential for healing wear and tear diseases.
The longevity connections for GSTA4 are incredibly robust across the board. When researchers analyzed huge multi-tissue databases of interventions known to extend lifespan,
Things like caloric restriction, the immunosuppressant drug rapamycin,
and diabetes drug, metformin, and even genetic growth hormone receptor knockouts.
GSTA4 is consistently upregulated in all of them.
In fact, it ranks in the top 0.71% of all genes associated with maximum lifespan extension in mammals. It isn't just a side player.
No, it is a fundamental gear in the machinery of longevity.
Hearing all this, you might be wondering how you can actually benefit from this pathway right now without signing up for experimental gene therapy in your eyes or your joint.
Right, because that's not exactly available at the local pharmacy.
Not quite. But the sources provide a very practical clue involving an over-the-counter supplement called Glynas, which is a combination of the amino acid glycine and N-acetylcysteine.
To understand why Glynax matters, we have to look at how GSTA4 actually does its job. GSTA4 doesn't magically destroy the toxic 4-HNE exhaust all by itself. It detoxifies it by conjugating it, which means binding it together with a molecule called glutathione.
Glutathione is one of the body's master antioxidants. The problem is, as we age, our natural levels of glutathione plummet drastically.
Which means even if you have GSTA4 present and ready to work, it doesn't have the raw material it needs to do the job.
Linacy supplements provide your body with the exact raw precursors it needs to rebuild its glutathione supply from scratch.
In the laboratory tests outlined in the 2025 paper, researchers found that giving Glynaceae robustly protected RPE cells from oxidative death.
But crucially, it only worked if the GSTA4 enzyme was present to use it. Supplying Glynaceae is a direct, actionable way to support this exact anti-aging oxidative resilience pathway.
It's a very clear, practical application of all this dense biology.
So, to summarize the incredible ground we've covered in this deep dive, we started with a mysterious 2003 observation about mouse cells essentially hyperventilating and dying of oxidative shock in standard lab petri dishes. That fundamental realization about oxygen paved the way to a 2025 breakthrough where scientists are using a dormant embryonic detox enzyme, GSTA4, to clear out toxic cellular exhaust, restore vision, and literally cut the biological age of eye cells in half.
And we are seeing early signs that this exact same pathway might be a master key to healing joints and promoting whole-body longevity.
It is just phenomenal.
But, this raises an important question, one that fundamentally challenges how we conduct preclinical medical research.
Oh, absolutely.
Think back to our very first source. If mouse cells are so catastrophically sensitive to 20% atmospheric oxygen that it literally causes the massive DNA damage and premature aging in a petri dish,
Yeah.
how many thousands of promising drugs, potential cancer treatments, and longevity interventions have failed in the laboratory over the last few decades simply because the mice they were tested on were breathing standard atmospheric air?
That is wild to think about.
Does the entire foundation of preclinical medical research need a massive oxygen adjustment to truly mimic human biology and give these therapies a fair chance?
That is a staggering thought to leave on. The idea that the very air in the lab could be altering the course of medical history. We hope you enjoyed this deep dive into the hidden wars happening inside your cells. Keep asking questions, stay insanely curious, and we will catch you on the next deep dive.